Hello everyone. In this class, we'll try to learn about gastric motility. The objectives would be name the gastric motilities, appreciate the difference in motility in proximal part and distal part of the stomach, correlate functional modification in stomach wall to carry out gastric motor functions, understand the importance of innervation of stomach, elucidate the electrophysiological gastric smooth muscles, outline the types of gastric relaxation and their importance, describe mechanism and regulation of gastric emptying, comprehend the electrophysiology of gastric contraction with gastric motor dysfunction, to learn the mechanism of omitting. Stomach is divided into two parts, the proximal part which acts as a reservoir and the distal part which acts as a pump. The proximal stomach, for its property of receptive relaxation, receives and stores food, and its tonic contraction pushes the food towards the antrum. The distal part, for its property of phasic contraction, mixes, grinds, and breaks down the food into smaller particles and regulates gastric emptying of food into small intestine through gastroduodenal junctions. The major objectives of gastric motility are to allow the stomach to act as a reservoir for storage of large amount of food of a single meal and to convert it to chyme and allow gastric contents to enter the duodenum at slow but controlled rate. See the stomach, it has a proximal part, consists mainly the fundus, the major part of the body of the stomach. The proximal part of the stomach accommodates large volume as large as 2 to 4 liters without much increase in the intragastric pressure. This becomes possible due to the phenomenon of receptive relaxation. The contractile ability of fundus and body of stomach is normally poor. Therefore, food in the fundus and body remains relatively unmixed for longer duration. Thus, proximal stomach serves mainly as a reservoir of food. So this is the proximal part of the stomach which exhibits tonic contraction and relaxation, and this is the distal part of the stomach which helps in grinding, mixing and controlled emptying of the food. Okay, so it exhibits phasic contraction which is called as antral pump. The distal stomach consists of mainly the antrum. The antrum physiologically acts as a mechanical pump which propels food towards the pylorus and helps in grinding and mixing of food. The antral contraction breaks foodstuffs into smaller particles and mix food thoroughly with gastric juice that help in partial digestion of food. Pyloric sphincter remains partially closed and does not allow easy entry of gastric contents into duodenum. However, vigorous contraction of gastric antrum help gastric content enter into the duodenum at slow but controlled rate. After grinding and mixing of food with the gastric juice, the gastric content is now called chyme. See the structure of the stomach wall. All the general components of the wall are present. However, the circular muscle layer of the muscularis externa is more prominent than longitudinal layer. In general, muscularis externa in fundus and body is thin, and antrum and pylorus is considerably thick. The thickness of the muscularis externa increases from body towards pylorus. The highly developed muscle coat helps the antrum to work as an effective pump. So this is showing the different layers of the stomach wall, fundus, body of the stomach, antrum of the stomach. Okay. Note that the muscularis externa is more developed in the antrum than in the fundus. The junction between stomach and duodenum is called as gastroduodenal junction. The pylorus separates antrum and duodenum. There is a sphincter in pylorus consisting of a ring-like thickening circular muscle fibers is known as pyloric sphincter. So this allows the gastric content to enter into the duodenum at a slow and controlled rate, the rate at which the duodenum is capable of processing the chyme. and to prevent regurgitation of duodenal contents back into stomach. Gastroduodenal mucosa is relatively resistant to bile acids but sensitive to gastric acid. In incompetent pyloric sphincter, regulation of duodenal content, that is containing bile acids, into stomach usually results in gastric ulcer. On the other hand, rapid emptying of gastric contents containing acidic chyme into duodenum promptly causes duodenal ulcer. Normally, as soon as acidic chyme from the stomach enters the duodenum, the acidic pH stimulates the release of secretin from S cell of duodenum in upper jejunum. Secretin stimulates pancreatic secretion rich in water and bicarbonate. Therefore, immediately acidic chyme is neutralized by aqueous component of pancreatic juice. However, when gastric emptying faster or secretion of pancreatic juice is less, the possibility of duodenal ulcer is more. Moreover, the content is in the part of the duodenum above the sphincter of Oddi, not get well mixed with the pancreatic secretion. Neural control of the pyloric part. The pylorus is richly innervated by parasympathetic and sympathetic fibers. Normally, sympathetic stimulation causes constriction of the pyloric sphincter, and activation of parasympathetic has a both stimulation and inhibiting effects. Simulatory effects are mediated by cholinergic vagal fibers. Stimulatory effects are mediated by the cholinergic vagal fibers. Inhibitory effects are mediated by vagal fibers that release VIP or nitric oxide. Constriction of the pyloric sphincter also occurs in response to hormones like CCK, gastrin, GIP, and secretin. Therefore, these hormones slow gastric emptying. Innervation of the stomach. Parasympathetic innervation stimulate, whereas sympathetic innervation fibers originate from celiac plexus inhibit gastric motility and secretion. Axons arising from intramural plexus innervate smooth muscles and secretory cells. Sensory fibers from stomach travel to CNS via vagal and sympathetic fibers. Some of the sensory fibers act as an afferent link between sensory receptors of gastric mucosa and intramural plexuses. Few of these afferent fibers provide information about the intragastric pressure, gastric distension, chemical composition, and pH of gastric content, and pain sensation originating in the stomach. All these stimuli influence gastric motility. The peristaltic wave in the stomach occurs at frequency of gastric slow waves. These peristaltic waves are generated by a pacemaker zone located in the middle of the body of stomach. The frequency of peristaltic wave is about 3 per minute in human being, and the waves are conducted from body towards pylorus. The gastric slow wave has 4 phases that resemble the action potential of cardiac muscle. However, it does not overshoot and lasts for a longer period, 10 times that of the cardiac action potential. The smooth muscles of stomach contract when depolarization of the slow wave exceeds the threshold for contraction. The force of contraction depends on the degree, frequency, and duration of depolarization. Greater the depolarization, longer the muscle cells remain depolarized, that above threshold, greater the force of contraction. In the antrum of the stomach, action potential spikes occur in plateau phase. The contraction that results from these action potential are stronger than the contraction that occur in the absence of these action potentials. Acetylcholine and gastrin improve gastric contractility by enhancing the amplitude and duration of the plateau phase of gastric slow waves. Norepinephrine inhibits by opposite mechanism. Types of gastric motility. Peristalsis of the stomach results in regulated gastric emptying. Hunger contraction. When stomach is empty, motility of the stomach increases. If the stomach is allowed to remain empty for a longer duration, the contraction becomes vigorous. These contractions are called as hunger contractions. Usually, hunger contractions are distressing and painful. The antral contractions are intense in such a situation and are associated with relaxation of pyloric sphincter. Gastric relaxation. initial receptive relaxation. This is relaxation of the fundus of the body of the stomach in response to chewing and swallowing of the food. Because of the receptive relaxation, intragastric pressure does not rise. The receptive relaxation is mediated by vagus nerve. Normally, gastric motility induced by vagal stimulation is mediated by cholinergic fibers. It is mediated by vagus is due to release of neurotransmitters like VIP and nitric oxide, that is non-cholinergic fibers at its nerve ending. So this is a diagram showing the receptive relaxation when the food enters into the stomach. So the stomach fundus is going to dilate, relax. Adaptive and feedback relaxation. Two types of gastric relaxation, adaptive and feedback. The adaptive relaxation is the relaxation of the stomach triggered by distension of the stomach. The receptive relaxation starts even before food reaches the stomach, where adaptive relaxation occurs in response to stretching of the stomach wall. This is mediated by vagovagal reflex, and it is meant to accommodate a greater volume of food. The feedback relaxation of stomach is a reflexive relaxation that occurs due to the presence of food in proximal segment of small intestine. So acidic chyme and fatty acids in the intestine inhibit gastric motility by both hormonal and neural mechanism that cause feedback relaxation of the stomach. So this is a flow chart showing the mechanism of relaxation, that is receptive, adaptive, and feedback mechanism. Okay. So when there is a chewing and swallowing, okay, so they stimulate the medulla vagal nuclei, and that leads to a receptive relaxation, where the entry of food in the stomach, gastric distension, um, and then the gastric distension, the acidic chyme and the fatty acids uh, in the intestine, they cause the feedback relaxation of the stomach. Okay. And even sometimes entry of the food in the stomach, I mean by vagovagal reflex, so it is going to cause a receptive relaxation of the stomach. After about half an hour following gastric filling, gastric peristalsis starts. The peristaltic movements are initiated by gastric slow waves. The pacemaker for gastric peristalsis is located in the middle of the stomach, close towards the greater curvature. It starts as a ring of contraction that progresses slowly towards antrum. It usually occurs at the rate of 3 per minute. It helps in propelling food into antrum and mixing of food with gastric juice. During this gastric peristalsis, pylorus usually remains closed. Therefore, food does not enter the duodenum, rather it is reticulated in the stomach, recirculated in the stomach. Migrating motor complexes. During interdigestive phase, antrum of the stomach remains silent for about 75 to 90 minutes, after which a burst of electrical and motor activities occur. This causes intestine antral contraction with the relaxation of pylorus. Therefore, the remaining gastric content is emptied into the duodenum. It helps in the cleaning the stomach and intestine. Reverse peristalsis. Sometimes in abnormal situation, peristalsis occur in reverse direction, which starts in the lower part of the body and proceeds towards the esophagus. The lower and upper esophageal sphincter relax so that gastric content is forced out of the esophagus and oral cavity. So this results in omitting. Gastric emptying is the process by which the content of the stomach is emptied into the duodenum. Usually it occurs at slow but controlled rate so that duodenum uh, and jejunum receives at the chyme at the constant rate. Mechanism. When food enters the stomach, stomach relaxes due to receptive and adaptive relaxation. Later, after about half an hour, slowly gastric motility begins. Gastric emptying occurs by three mechanisms. One is peristaltic contraction, antral contraction, and retropulsion. Peristaltic contraction. The peristaltic contraction usually begin in the middle of the stomach and proceeds in a ring-like fashion towards pylorus. These contractions mainly push food into antral part of the stomach. Velocity and magnitude of contraction increase as the contractile waves approach pylorus. As the contractions are weak in the fundus and the body of the stomach, the proximal portion of the stomach mainly serves as a reservoir function. Therefore, usually proper mixing of food with gastric secretion does not occur in these parts of the stomach. Major amount of mixing takes place in the antrum as the antrum contracts vigorously. Antral contraction help in thorough mixing of food with the gastric juice. The forceful contraction of antrum forces gastric contents towards pylorus. But as the pyloric sphincter remains closed, peristaltic wave fails to push food into duodenum, rather food returns back into the body of the stomach. After few such contractions, pylorus opens partially with narrow opening at the center. Therefore, stomach empties in a small squid with each peristaltic wave. Retropulsion, it occurs in the terminal part of the antrum where when rapid and forceful contraction that forces the chyme to be propelled back to the proximal part of antrum and body of the stomach. So retropulsion is very effective in mixing and grinding of the larger food particles into smaller ones. Okay, so these are the mechanisms showing the gastric emptying, peristaltic contraction, antral contraction, retropulsion, and antral pump. As the muscle layers in the fundus and body are thin, the contraction in these part of the stomach are weak. Therefore, gastric content in the body of stomach settles into different layers based on the density. Fat content of the food forms oily layer on the top of the gastric contents. So this is why fat is emptied slower than the carbohydrate and protein. Liquid portion of the food flow around the mass and enter the antrum and from there into the duodenum. Therefore, liquid is emptied faster than the solid. Regulation of gastric emptying. Gastric emptying is regulated by both neural and hormonal mechanism. Acid in the duodenum, which with a decrease in the pH of the duodenal content, the rate of gastric emptying decreases. This response is mediated by both neural and hormonal mechanisms. The acidic chyme in the duodenum releases secretin that decreases gastric emptying by inhibiting contraction of antrum and by stimulating the contraction of pyloric sphincter. Products of digestion. Products of fat digestion like fatty acids and also some of the fat molecules in the duodenal content inhibit gastric emptying. This response is mediated by CCK and GIP. CCK is secreted from duodenum and jejunum in response to fatty acids and it inhibits gastric emptying. GIP, which is released in response to fatty acid and also inhibits gastric emptying. Osmolality of the duodenal contents. The chyme entering into duodenum has a higher osmolarity. The osmoreceptors in the mucosa of duodenum and jejunum that detect change in the osmolality of the duodenal content. The hypertonic solution in the duodenum release hormones that inhibit the rate of gastric emptying. The products of protein digestion. Presence of peptides and amino acids in the duodenum release gastrin from G cells located in the duodenum. Gastrin increases antral contraction but at the same time also causes constriction of the pyloric sphincter. Therefore, the net effect is decreased rate of gastric emptying. The product of protein digestion also release CCK and GIP from duodenum and jejunum that inhibit gastric emptying. Volume of the meal. Whenever the large amount of food is taken in a meal, the time taken for gastric emptying prolongs. However, if the volume is mainly due to the liquid, then emptying is faster. Stretching of the duodenum. Entry of the chyme into the duodenum stretches the wall of the duodenum. So this initiates enterogastric reflex that inhibits gastric emptying. Enterogastric reflex is also activated by acid in duodenum. Neural factors. Vagal stimulation promotes gastric emptying. Therefore, vagotomy reduces or produces gastric stasis. Consequently, whenever vagotomy is performed for the treatment of peptic ulcer, usually a drainage procedure like pyloroplasty or gastroduodenostomy is also performed to ensure the proper passage of food from the stomach into duodenum. Sympathetic stimulation inhibit gastric emptying. Hormonal factors like most of the hormones liberated from the duodenum and jejunum like CCK, GIP, secretin, etc. inhibit gastric emptying. Coming to some of the applied aspects, dysfunctions of gastric emptying, that is, in case of delayed gastric emptying, the gastric emptying is delayed in autonomic neuropathy. Paralysis of propulsive movements occur following vagotomy, which is called as gastroparesis. Hypertrophic pyloric stenosis can cause gastric stasis. Rapid gastric emptyings. Normally, vagus stimulation promotes gastric emptying. Therefore, states of increased vagal activity increase gastric emptying. Conversely, sympathetic stimulation inhibits emptying. Therefore, loss of appetite is a feature of uh, stress, a state like sympathetic overactivity. Hormones like thyroxine stimulate gastric emptying and intestinal motility. Increased liquid content of the food increases gastric emptying. Omitting. It is the expulsion of the gastroduodenal contents from GIT to the external environment via mouth. Associated features it is usually preceded by feeling of nausea, tachycardia, sweating, pallor, dizziness, and dilatation of the pupils. It is associated with retching that forces the contents from stomach into the esophagus. What are the stimulus? Omitting is a reflex phenomenon, the center for which is located in the medulla. The receptors present mainly in the part of the body provide inputs to the omitting center in the brainstem. Omitting center is present in reticular formation of the medulla and consists of various scattered groups of neurons that control different aspects of omitting. Vestibular nuclei mediate omitting in response to motion sickness. Pharyngeal stimulation induces omitting by activating nucleus tractus solitarius. Area prostrema mediate omitting activated by drugs and hormones. Omitting activated by emotion is influenced by limbic and diencephalic inputs. The important stimuli that activate these afferent fibers are distension of stomach, tickling the back of the throat, painful injury of the genitourinary tract, conditions associated with dizziness and nausea, many chemicals or drugs that can elicit omitting. The drugs that induce omitting are called as emetics. The common emetics are apomorphine, ipecac, etc. These chemicals stimulate the receptors that are present in the CTZ, that is chemoreceptor trigger zone, located in the area prostrema, a V-shaped band of tissue on the lateral wall of the fourth ventricle. Many anti-emetics prevent omitting by inhibiting CTZ. So there are 5-HT3, that is serotonin, and D2 receptor in area prostrema. Serotonin stimulate through the 5-HT3 receptors, and Wodensetron 5-HT3 receptor antagonist is an anti-emetic. Chlorpromazine is a D2 receptor antagonist, and haloperidol also used as effective antiemetic. Corticosteroids, cannabinoids, benzodiazepines are useful antiemetics for chemotherapy-induced omitting. Cases of reverse peristalsis that starts from middle of the jejunum. This sweeps the content of intestine and duodenum into stomach. Second, relaxation of the pyloric sphincter that allows the intestinal content to enter the stomach. Third, forced inspiration occurs against the closed glottis. Lowering of the diaphragm increases the intra-abdominal pressure. This is followed by the vigorous contraction of the abdominal muscles. Thus, intra-abdominal pressure sharply increases, which forces the gastric content to enter the esophagus. Relaxation of the lower esophageal sphincter allows the gastric content to enter into the esophagus. Normally, the upper esophageal sphincter remains closed and prevents regurgitation of food into pharynx. With a stronger retching and sharp rise in intra-abdominal pressure, the upper esophageal sphincter relaxes, which allows chyme to enter the pharynx and then from there into the mouth. Entry of omittus into trachea is prevented by central movement of the vocal cords, closure of the glottis, and inhibition of respiration. So this is showing the mechanism of omitting reflex. Okay, so visual inputs, limbic inputs, cortical inputs, vestibular inputs, they trigger the chemoreceptor trigger zone that leads to omitting. Otherwise, the touch receptors in the throat and pharynx and the input from the stomach and the intestine because of the gastric distension and chemical stimulation can stimulate omitting center. Okay, so from the omitting center, the efferents goes to the upper GIT, that is 7th, 9th, 10th, and 12th cranial nerves, and the spinal nerves to the respiratory and abdominal muscles. So, which leads to expulsion of the gastric contents to the exterior. So, which is called as omitting. Dumping syndrome. This is a distressing syndrome consisting of mainly weakness, dizziness, and sweating that develop in about 2 hours after meal in person who have undergone gastrectomy or gastroduodenostomy. So this occurs because of two causes. One is hypoglycemia. Rapid entry of food into intestine causes quick absorption of glucose from intestine and produces prompt hyperglycemia, in which in turn increases insulin secretion. Insulin produces hypoglycemia that produces weakness, dizziness, and sweating. Second cause is hypovolemia. The speedy entry of a hypertonic meal from esophagus directly into intestine produces high osmotic load on intestine that transfers water from blood into the gut. So this produces hypovolemia, dehydration, and hypotension that leads to weakness and dizziness and sweating. So this is in brief about the gastric mobility and gastric motility and some of the applied aspects related to it. Thank you.